Okay, we are
So,
live.
good evening, everyone. My name is Adikemi, and today I'll be taking us data format. So, first of all, I'd like to apologize because I'm a little bit under the weather, and I'll try my best to take us through this topic. So, for some of us, I know the word. I hope everyone can hear me. Please, can we all hear me?
Yes, I can hear you.
So, so as I was saying, I forgot what I was saying. For some of us, I know that the word format or file format might be like, oh, okay, what exactly are we talking about? Um, I want us to think of it as learning the language bioinformatics speak. And when you learn the language it speaks, you'll be able to understand what the data is saying. And also, it's a very key thing for doing great analysis. If I use myself as an example, when I just started, though we are always learning, seeing different formats. I would say I was like a bit intimidated, especially when you see a lot of A's, you see a lot of T's, G's, and C, and you be like, "Ah, what exactly am I even looking at?" So, when you know the formats, you learn how the data is structured. And so basically today, we'll take a tour through the world of bioinformatics, and then we'll start from where they are from, why they exist, and go through major data formats, which we'll go through afterwards, like the FASTA, the FASTQ, the SAMBAM, PCF, GFF. So, and hopefully by the end, you are able to recognize each of these formats. So let's go straight in. So what is a data format? We can say a data format is like a set of rules. It defines how the data is organized, stored, and how computers can interpret it. So it's defined by the information it contains, the design and optimization of that information. I'm sure a good number of us, we watch movies, and you see that you can have movies in different formats. You see some of them end with MP4, some AVI, MK, even pictures. You see some with JPEG, iPhones with HEIC, different formats. So it's like the same story. And we know that format can affect the size, can affect the quality. Using the example of movies that I mentioned, and how easily different devices can play it. You find out that some phones can play this format and some phones can't play this format. So in bioinformatics, it's the same thing. So you have your DNA sequence data, you have your protein and mutations, etc. And each of them, they need to be stored in a format that best fits the purpose. So when we talk about format today, just think of the file name. You can think of your design choices. Why did somebody do this? Or like, why did somebody make this format? What are we trying to ask? What are we trying to solve? Because when you know these questions, you'll be able to know, oh, okay, which format am I supposed to use for my research? So I'm sure we've seen, just like I mentioned, or maybe not, we've seen dot faster, we've seen dot fastq, we've seen dot bam file before. So if you check it, it might look very gibberish, but once you understand the logic behind it, then you know, okay, this is the purpose. So it's like, yeah, cracking a code. So I hope I'm not saying too much story. So sequence data storage evolution. So let's go back to the early days of bioinformatics. So right in the beginning, sequence data was stored in the simplest way possible. And that's like plain text. literally you see .txt. So files full of sequences. You see like maybe you see your nucleotides, your AT, GCC, then in .txt. So, and we are talking about evolution now. And back then that was enough. We didn't have as much, or we didn't have high powered computing devices. I hope I'm correct with that. Like computers were slower, um, data sets were small, and we did not have as much massive sequencing as we do today. So, but as time went by, then we realized, just having sequences is not enough. Oh. We also need to know where it comes from, which gene it's part of, what organism, how reliable is this data. So imagine I said it was in .txt. So .txt, or let me say plain text, just could not undo that. Is like you have how should I put it? It's like having a book with no chapter titles, nothing, just letters everywhere. So then people started coming up with new formats to hold more complex information, as we as science became more and more advanced. So that's where you have things like GenBank, FASTA coming into play, and each of them having more structure and more meaning. And then we have, I'm sure a good number of us have also heard of the, I hope everyone can still hear me.
Yes.
Okay. And then I'm sure a good number of us have heard of the next generation sequencing. And, this totally changed everything. We're not talking about just maybe hundreds, or should I say few thousand sequences. We're talking of millions of reads, each with its own quality score. And then you see, then we came up with FastQ, SAM, BAM, VCF. So basically, in short, is like as sequencing evolved, as sequencing technology evolved, as everything started changing, so did the file format from the TXT, from our plain letters to different data structures. So if you look at this tree here, you see that we have so many data formats. You can see your FASTA, FASTQ, GenBank, SAM, VCF, etc., etc. So why do you have this many formats? Is like, can't we just use one? Different, the answer would be that different tasks require different levels of detail and structure. So as you can't use, I'll try to use as much analogy as well. As you can't use maybe one recipe to make different food, the same thing applies to the data format. So everything comes from different angles. So for instance, now your FASTA will give you, your FASTA file will give you the raw sequence, the sequence rather, and then your SAM and BAM, they will show you where it fits, at it here, sequence alignment, etc. So each format is optimized for different tasks, every single one, as you can see here. So faster for your storage of sequences, your FASTQ for sequence, your quality information, your SAM-BAM for alignment of reads to a reference, your DCF for storing genetic variants, your GFF describing gene structures and features, so gene annotation, So every single one of them is doing different things. So once you know what each one is for, then you know that all of them are complementary. You can't just jump from one place to another. Do you understand? So it's like if I put, I can say it's like an ecosystem of data. So okay, before I go to Gembank, so you know, as we go through all the different formats, you see how we're able to like read each of these formats, because hopefully when you open one you'll be like, oh okay, this is what this is what that is, oh this is fast speed, this is faster, and all. So Gembank is like the grandparent of data format, and we can think of, it's one of the oldest bioinformatics data format. So you can think of it as an encyclopedia. So it was created to collect, to store, and to share DNA and protein sequences. Also, even in fact, GenBank is one of the first attempts to organize biological data. And then it is still, if you check NCBI, which is the National Center for Biotechnology Information, you still find GenBank. It's still maintained by the NCBI and is the backbone of a lot of many other formats. So the idea was very simple. Oh, okay, make a format that is both human readable and machine readable. So, you know, especially then, when I said computers were not as advanced, most of the data was written for people. So you see, they were printing out sequences. So if you think of GenBank, you can think of it as like an early database language. So looking at the structure, so GenBank actually ends with JOT. When you see .gb, you know it's a GenBank format, and it has 5. If you look at the diagram here, you can see the you can see these structures. You can see locus, definition, assertion, version, etc., etc. So the locus here, it gives you the name of the entry, what type is it? Is it DNA? Is it RNA? Is it protein? And also, even when it was last updated, if you look at this now, it was last updated April 10, 2023. And your definition gives you like a small or a short description of, okay, what is this? Oh, this is Arabidopsis thaliana, blah, 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 like that. And you also have the accession number. Then you have the origin here, so which is the actual sequence itself. So usually when you see the origin in GenBank, it also comes with, you see the sequences itself. And though you can't see it in this screenshot, usually it ends with double slash. So once it ends with that double slash, it means that is the end. So, and if you look, and even the screenshot itself is not even showing you like the full details, because GenBank can be very, very heavy. Like you have, like, can be very text heavy. You have a lot of information in it. So, and one of the most powerful thing about GenBank is that it can store metadata. So you have like all the description, like the ones we went through the other time. So you have the organism name, you have the taxonomy, even it will even reference some other databases as well, even literature references, etc., etc., like that. The The features as well, it will show you the coding, the CDS. I don't, it's not here, right? So if you go to GenBank, you will find, you see the coding sequences, you see something like gene, blah, 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 product, all of them. It will even give you coordinates. So many information that you find in GenBank. And the not not optimized for analysis. One of the limitations of GenBank is good for storing data, but if I should put it this way, in my own terms, I would say it's not perfect. It is not for analysis. It's too big. It's not particularly fast for computational processing. So that's why you see that we often convert GenBank files into simpler file formats. And if I ask somebody, well, I don't know if you give me the answer, but you see simpler formats like faster formats. So another limitation is that GenBank is human readable. You can see that these are all text telling us the authors, the references, all of those things. So all the how should I put it now? So handling massive data set in GenBank is not very efficient. So if I say think the GenBank is like, oh, okay, it will give you everything. Exactly. Let me just put it that way. The GenBank will give you everything. It will give you, using an example now, the GenBank will give you the note. Let's say the architect now, the note, the history. If you use a house, for example, it will give you, oh, okay, this is the architect's note. This is the history of the house. This is the person who painted Tutuku's room. This is the person who brought out this part of the house, all those things. But the next format, which we'll be going to now, which is the faster format, it will give you, what it will give you won't be all those stories, but it will give you the structure. It will give you the rooms, the walls, not all the other information you don't need. I hope we're still all on the same page. Please, can you people
Yes,
see?
yes, I can see thumbs up.
Oh, okay. I can't see the screen. Sorry, that's why. So, like you think of faster, like a simpler version of GenBank. So which is what we'll be going to now. So we are done with GenBank, which I mentioned is like the grandfather, grandmother, grandparent, yeah. So now I'm sure a lot of us have heard faster files everywhere. When you want to do your blast, your reference, you want to do kiniko, you are dealing with faster files. So it is very, I would say that it is very simple that sometimes we can even underestimate how powerful faster can be. So what exactly, sorry, what exactly is faster format? So it's the workhorse of bioinformatics. Can say it's a text-based format that is used to represent either DNA, RNA, or protein sequences. So every faster you see always starts with a greater than sign. So if you look at my cursor here, you can see greater than. So once you open a format and you see greater, the header starts with the greater than sign or the greater than symbol, then you know that it's a faster format, and it's usually followed by like a short description here, like you can see here, and then the sequence. So basically one header here and then the sequence. So This header will actually give you the context. Here you can see Arabi no Jesus Christi, Arabidopsis thaliana, the gene, the organism name, the accession number, and then you can see the raw biological data. So okay, so file extension. Sometimes, well, faster files usually can end with .fa, can end with .faster. You might even see .fna. That one is for nucleotide faster, or even FAA. That's for amino acid. So all faster files. So it's a way for it's a naming convention that helps people and computer to recognize the type of sequence inside. So basically, you can name your file anything. You can name it anything, but it's a if how should I say it? You can name your file anything, but make sure that it ends with the correct um, file format. So if I go to versatility of FASTA. FASTA format is not limited to DNA. If you remember, I mentioned RNA, I mentioned proteins and stuff like that. So where each letter corresponds to an amino acid, so etc. etc. And before we even go to the FASTQ, why we should think, okay, why is FASTA file so widely used? FASTA is very universal. You know, if you remember, I mentioned that it is a simpler version of GenBank. It's simple and lightweight, and it doesn't try to do extra things. It's just, oh, okay, I'm just storing sequences. So the because it's simple, it makes it easy to write script. You can use Python, you can use Bash, R, you can use it to extract things. You can use it to extract length, you can use it to rename and so on. So, but faster faster files, they do not contain quality scores, or let's say sequencing information. It only knows the sequence, and which we can say is good. But once you start sequencing a lot or millions of reads at a time, you need a format. You need a format that can store how confident we are in each base call, and which is what is going to bring us to the FASTQ format. So FASTQ format is one of the most important formats you deal with. And I would say that FASTQ is the language of sequencing machines. So it's the most common output from the NGS platforms. So FASTQ is a text-based format that includes sequence identifiers, the sequence itself, an optional plus sign, and excuse me. So they are like what comes straight off from the NGS platform. So they don't just contain the DNA sequences. They tell you the quality score for each base. So it usually has 4 lines. So here, if you look at the screen here, you have, this is the sequence identifier. Starts with at so so so. So read sequence identifier. The second line is the sequence itself, and the line 3 is plus, which can be optional. Some it can be optional, but it is a separator, this plus sign. And then this line for might look gibberish, but it's not how should I say it? It's not weird. So they actually tell us the quality values and how reliable each call is. So they are string of these characters. What do we even call this? I don't know if I'm to whether I should say ASCII. That's what I might be wrong in the pronunciation, but this basically. So there are string of these characters. So imagine now, let's say T, like every one of us, you go to classes, you write an exam, or for instance, the test you have to write or you've written, you got 40. So let's say maybe this is Abib, this is Tochukwu, this is Wanokwe, this is Kemi, this is, and each of us we now have, I have 80, he has 40, they have 80. Imagine having to write that numbers one after the, it won't fall into place. So all these characters here actually represent a score, a value. I think, okay, which we'll see here. I'll show us. So this fourth line, like I mentioned, they contain what we call Fred scores. So what those scores are called Fred scores. And the Fred scores are like numerical values that measure the probability that, oh, okay, this that you called was called incorrectly. So if you go to the, okay, this next slide actually shows us how it's being calculated. So the formula is error probability is equal to 10 raised to power this over 10. So it's like if you have a score of, okay, before we even, okay, so let me just show us here. For instance, now this, okay, this colon here, or this 0, for instance, if you look at it, a Fred score of 0 is indicated by this exclamation mark. So that means, let me see, can we see? Oh, okay, good. There's no, because look at it, it's going from worst to best. So the highest Fred score you can have is 40. So anything, if it's going downwards, it is bad, badder, baddest. So thank God for this picture. There's no exclamation mark of 0. Okay, looking at the one that was being highlighted here, it's a colon. So that means it has a Fred score of 25, which is good, not bad. So yeah, I think there's a mistake here. It's 30 for 5. I don't know. So, but basically, you just understand what I'm saying here. So each of these symbols you are seeing, they actually mean something. So going back to this probability, so if you have P is equal to 0, that means 100%. Or remember, look at it, worst. So that means 100 out of 100, the base call is called incorrectly. So if you have a score of 10, it means you have 1 in 10 chance of being wrong. So that's still a lot, 1 in 10. If you have a score of 20, it means 1 in 100 chance. Still a lot, actually, but fairly better than 10. So so now look at this, which we said is the best. P is equal to 40. So it means you have 0.01% error chance. So 1 in 10,000. So that is very, very good. So the higher the Fred score, the more reliable the base score. So, and what we want is that we want to reduce error as much as possible. So when you see this, you know they actually mean something. These are, you get your Fred scores. So yes, I hope I was being clear then. So sometimes when you see some medals, it might look like what's it called? Chaos. And in this instance now, you can see this EAS colon this. And it might look gibberish, like we've been saying, but everything has a meaning. So here, this is the instrument name. This actually indicates the run ID. So this is from Illumina. So because sequencing, so if you're wondering, okay, we're just saying this. So once you have this format, so here now, this is the instrument name, the run ID, the flow cell. So this diagram actually shows this is the flow cell. You can see a lot of partitions. So this indicates the cell lane. All of this basically they have a meaning. The one this actually shows the x and y coordinates of the cluster within the tile. So one, if it's just a member of a pair. So two, if it's pair end. So y here, if the read is filtered. So let's just simplify this example that we are being shown here. So it's basically saying that the machine EAS139 did a run with this run ID 136 using the flow cell this, and this particular read came from lane 2 tile 2104. Already showed the diagram. At position, this is the x coordinate, and then the y coordinates on the flow cell. It is the first read. Remember I said if the member of a pair, so it is the first read in a pair edge sequence. So the read quality checks, I mean the read field quality check. So y, so if you see y, it means the field quality check. And then the sample was identified by the barcode index. So this is the samples barcode. So imagine you are what's it called? You are sequencing, or you could sequence many samples at the same time. So each of them, they have their own barcode. So for this is its own barcode. So this actually is very helpful because you can tell, oh, exactly which or where in the sequencing machine did this read come from. So it's like you have a GPS for your DNA. So what is FastQC? Quality score. So basically, before then, please, are we still together?
Yes, I can see some thumbs up.
Okay.
But just two people actually. How about the others? Yeah, more thumbs up. Okay.
Okay. Okay. But before I go on, please, I hope I'm not losing anybody. And I hope I'm not losing myself. Is there thumbs up? I can't see the screen, so I don't know.
Yes, we still have more.
Come again?
Yes, there are still thumbs up. Yes, we are.
Okay.
I think we are good.
Okay. Thank you. So, um, first of all, why does quality score even matter? Like now, you've mentioned Fred score. You said, so let's use an example. There's a Fred score of 40 and there's a Fred score of 50. If you see 40, you are much more confident. You are much more confident than the one that has the Fred score of 15, or let's say 10. So, and you want to do downstream analysis, you want to do further analysis. Which one are you going to use? You have definitely going to go with the one with the higher Fred score. So because remember, you are good, you are just like I said, you want to do downstream analysis. A very low quality read would give you false many things, a lot of false. So and when I mean a lot of false, meaning if you do your alignment, you do variant calling, a lot of downstream analysis is going to be false, basically. So which is why we need tools like fast QC, and it is used before you do any major analysis. So you want to check, you want to summarize, you want to visualize your quality across all the reads. So um so what exactly is this fast QC? So it's like a checkpoint before you do the downstream analysis, just like I've said. And it will read your FASTQ files, which we just spoke about, and then give you a report. You can see some details here. So it produces some graphs for you. Here you can see per base sequence quality, you can see per sequence quality score, per base sequence content. So information like that. So your fast QC will give you, to show you your overrepresented sequences. So see your basic statistics here. It says file name this, encoding. It will show you your total sequences, your total bases, how many of them were flagged as poor, sequence length, your GC content, etc. So now looking at this diagram here, how do we even interpret this? You can see different colors. You can see green, you can see orange, you can see red. So green, when you see when green actually means good. Once you see that, oh, okay, this would fall in this line. That's good. Orange is like borderline, like when somebody say somebody is big, like oh, it remains small for this person to cross the border. That means it's warning. You are the verge. And then red is red. We always assign red to danger. So same thing here. So if you look at this diagram here, you can see that the threshold for this green is 28. And you can see that the quality scores is very, very high. This is very, very good. Like it's very good, and then you are confident in it. Look at a lot of them are in this green color. I mean, not a lot of them, all of them actually. Now compare it to this one. This is terrible. Maybe when this person was sequencing, the person was sleeping. If you see your data like this, just go home and go and watch K drama, because this is very, very poor. Anyways, jokes aside, this second one, you can see that few, no, just this one is even in the green line, this one the border line, a good number of them in the red zone. So you can't use this needs to be trashed, basically. And so this is good data and this is bad data. Same thing applies to this one. A good number of them have quality score here, you can see. But if you look at this bad data, very wobbly. About 10,000 have quality, their Fred score here. Even just looking at it, you can basically tell so that this is good and this is bad. And same thing with the GC content. Does it not look like this person is going through electric shock? I'm trying to be funny. Anyway, so same, just like we've been mentioning. So and I want to also mention that mm, if it shows if your per base sequence shows a steady drop at the end here, is normal. Do you understand? Because it's the machine. Because if you look at it, you can see that little by little there is like a steady drop. It's quite normal. Because for Illumina, because the machine actually tends to lose accuracy over time. So, but if the quality drops sharply, like this one, it's a very red flag, which I already mentioned. So it could be that you have to resequence, it's a failed library prep, contamination, many things. You just have to go back to see, oh, okay, what could be wrong. So, the fast QC is like the easiest. It's like when you want to check, oh, okay, how sane is my data before I go further down the line and then realize that I need to start all over again. So now we are done with the fast QC. Okay. So now we're in the SAM BAM file format. So we've cleaned our reads. We've used fast QC to check. We are very confident that, okay, yes, these are high quality. Sure. Now we want to align them to reference genome. It's like doing puzzles. You find patterns. If you look at these reads now, you can see we're aligning them. It's like, just like I said, you're doing puzzles. You find patterns and align them to a reference. So you can see TTs, CTs, TTs, basically alignment. So when you do this, the results are stored in a SAM or BAM file. So your FASTQ will tell you, okay, these are the reads we have, and then your SAM bar format will tell you where they belong. So see, the SAM bar file format, it enables various downstream analysis, including gene expression, biodiversity studies, methylation analysis, and protein DNA interaction studies. So the basic structure for a SAM BAM file. So you have the read name, SAM flag. So the read name can also be called the query name. You have the flag. Further downward, there's a slide that talks a bit about flag as well. So we'll talk about it. So the reference name, the position, mapping quality, the SIGA string. There's also a slide that talks about, because these are two important ones, so we'll talk briefly on them. So on and so forth, like that. So this, looking here, the HD here, think it can be ignored. I'm not sure though. But then this VN here is the version number, and then SQ tells you the name of the reference file, the sequence file, the sequence, the SN, so sequence file, sequence name, and also length. So okay, further down we'll talk about them. So the SAM files header. So just like how we've been talking about the fast queue. So the SAM files also have a header section. So the lines start with at so. So for example, now in this case, now you can see at SQ, SN, CHR 14, LN, Kinikon. So now this SQ here is basically it defines like a sequence dictionary. So the SN here is the sequence name, which is chromosome 14, and then the length number, which is this 10734, so on. And then here we have PG. So the PG actually is a program. So it records the program. This I said SQ define. This is the sequence dictionary. So this one is the it records the program, the version, and the command used. Give me a few seconds, please. Okay, I am back. Sorry, I want to. Okay, so as I was saying, so the program is so the SQ. I said sequence dictionary. So you can see the sequence name, the chromosome, the length, and then here you have the PG, which actually records the program, the version, the command. So the program is basically like the the is used to generate the SAM file and perform alignment. So it's a tool like used to generate the file. So your PN is the program number. This VN here is the version of the tool. This CL is the command used, and so on. So this actually is very helpful in reproducibility. So you can always say, oh, okay, this was how this file was created. It was created with this version. This was the command that was used, etc. and etc. like that. So okay. So here it talks about the flag. So indicate alignment information about the read. So the flag column, if you look at this column, you can see 99 0 0 0 2064 147. It will just be on, it looks like random numbers, but it's actually packed with information. So it's a binary code that tells us how the read was mapped, how it was aligned. So for example, a common flag value is 99. Where is it again? Here. So which is made up of 64, 32, 2, and 1. If you add it all up, it will give you 99. But each of them, they have their meaning. So 64 here, it means this read is the first in the pair, read 1. 32, the read is part of a pair and its mates aligned in the reverse direction. 2, this read is part of a pair that's aligned properly. 1, this is a pair end. So basically all these numbers here, they actually have meanings. So for here we have 2064. So it's probably 1024 512 256. Though I don't know the exact numbers, but I don't have to do that myself. So but then what I'm just trying to say here is they it tells us, it's packed with information. So it tells us everything that shows us how the read was aligned. So mm, so one interesting property of flag values caused by a value of 1 indicating that a read was paired is that pair end. Oh, I'm not mute. You can still hear me, right?
Yes.
Okay, sorry. So basically what this note here is saying that one oftentimes when the SAM flags are odd numbers, they are pair end data. So Siga, if you remember when I was showing this, I said two important ones. So we've seen the flag, and then now we want to check this. So what he actually means is compact. I think I wrote this down. Okay, so compact idiosyncratic gapped alignment report. So I don't know if you need to know the full meaning, but that's the full meaning. But basically, Siga. So it tells you how the read align to the reference. So it uses letters to describe the operation. So M for match or mismatch, I for insertion, D for deletion, S for soft clicking, H for hard clicking, N for skipped region. So in this year, can see that said 14M. Oh, okay. So in this part now we have 14 matches. So that means in total 14 matched correctly. 1, 2, 3, 4, 5. To the reference genome. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Wait, what am I counting? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So 14 matches, 14M, and then so 6 match. So if you look at this, you see that 6 match initially 1, 2, 3, 4, 5, 6, and then 2 insertions, which you can see here, and then 8 matches 1, 2, 3, 4, 5, 6, 7, 8. So basically that's what this 14M, 6M, 2I, 8M is telling us about. And then same applies to these ones. So you can see here there is a deletion 3D. So that's what the SIGA. So the SIGA actually it tells you how the read align. So is there a match? Is there a mismatch? Is there an insertion, deletion, skipped region, so on and so forth. So and these details, they are essential, especially when you are analyzing indels, insertion or deletions, or even spliced alignment. So now from SAM files, we are going to the BAM and the BI files. So SAM files are text, so they are very, very big. So I'll just try to summarize what this page is saying. I will just summarize it. So they are very big, and usually we compress them into BAM files. So BAM files actually contain the same data, but it is in the binary format. So you compress the SAM files to give you the BAM files. And then once you have your BAM files, you can sort. So you can sort by the read name or by the coordinates, and then you can create a BAI file. And so SAM is like, okay, the alignment information in plain text, and then BAM is like the compressed version of SAM, and then BAI is like the index file for fast access. So for instance, now if you want to if you are looking for something at chromosome 7 position, um let's say position 10,000 to 20,000, you don't have to like maybe go through the whole file. The index itself will tell the program where to go. So that's your BAI file like that. So I hope that makes sense to everybody. So I can just briefly say what I just said again. So your SAM file is the text file, very heavy, so you compress it to the BAM file. So the BAM files is the compressed version of SAM, and then BI is the index file. So the BI actually gives you a fast access. So all of these files that we've been talking about, they're like let me say the backbone of NGS, next generation sequencing analysis. So from variant calling to gene expression to structural variant detection. So application, so once both sorted BAM and BI files are generated, they can be utilized for various downstream applications, depending on the type of sequencing performed and the scientific questions being addressed. So if you remember back to the beginning, I was telling you, I was saying that, different file formats, you'll be able to know depending on the questions you are asking, okay, which one am I supposed to use here and there and everything like that. So we can see that we just did not jump straight from this file format to everything is stepwise. Actually, we went from the fast QC, I mean the fast Q, faster, SAM, BAM, and BI. So the VCF. Okay, so when you after alignment, the next thing, or yeah, the next thing you want to do now is a genomic. The next thing you want to do in the genomic analysis is variant calling. So the variant call format is a standardized format for storing and reporting genomic sequence variations. So it contains meta information lines, a header line, and then data line, each containing information about a position in the genome. So okay, first of all, the output of VCF, or before I even jump to that one. Okay, just like I've read, it's a standard way to store SNPs, insertions, deletions, and other variations. So it's stored in the VCF format. We call it VCF format, or variant call format. So you have the meta information. So you have the meta information, you have the header line, and you have your data lines. I hope I'm correcting this. Yeah. So in the in this here, you can see, okay, which is also here, file format. It tells you the version, which is 4.0, the date, um software used, reference genome. You find all this here. So which is the meta information lines. So the meta information lines usually start with hash hash. So all this is the meta information line. So metadata, all the information, and then you have your header line. This is your header line here, and then you have your data lines. So this one are your data lines. I hope I didn't confuse anybody here. So these are your meta information, the ones with the hash hash, double hash, and then the what's it called? The header line usually starts with hash pro, and then you have your data lines. So So in this, so this is basically telling us the full meanings of some acronyms here. NS, number of, okay, number of samples with data. DP, total reach depth, allele frequency, ancestral allele, DB membership, app map membership. And filter. So this slides actually just broke them down to, but basically all the information you're seeing they are here. So you can see what we just spoke about here. So here you have the quality below 10, and then S50 here is basically telling you less than 50% of samples have data. So every information you see here, they're not just there just to make the file wordy. They all have meaning. So, and then when you see GT, genotype, GQ, genotype quality, DP, read depth, and then haplotype quality. So for instance, now in this one, you have 20, 14, 370. So I'm just calling every one of them. So it means that on chromosome 20, position 14, 370. So we found a SNP. So SNP identifier, we found a SNP, which that is a change from G to A, your nucleotide base. And the variance passed filters, you can see pass here, and was observed in 3 samples, and it has a depth. Remember, we said DP. Where's DP again? Oh, okay. Basically, what I'm saying is here. So, as a depth of 14, an allele frequency of 0.5. So, and this H2 here is saying database. The variant is already known in scientific database. So each of them, I mean, when you look thoroughly, you see that each file format is actually passing a message. So so we can see that the example we just used now is good. Like it's basically it has good confidence. The quality score is 29. So it passed all quality filters and occurs in a good number of the samples that were being analyzed. So that's like this shows like the beautiful of I say beautiful, the beauty of PCF. So it's a like a structured but easy way to understand some of the information it gives you. So and then just like I've been saying, you can see how these files link to one another. So from SAM to BAM to VCF, etc., etc. So now we are going to GFF, and GFF is known as the general feature format, and the GTF is the gene transfer format. So I would say so far we've looked at different sequence forms. We've looked at the alignment, the variant, blah 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 like that. So now we want to describe features. We want to describe features like genes. We want to describe features like exons, promoters, coding regions. So we want to describe this. So this is where the GFF and the GTF file they come into place. So it's a simple file. It is tab delimited. So it describes the genomic features, the positions, the strands. So So now for example, using this one, the CTG 1 to 3 gene, this and this. So it is basically telling you that um gene Eden running from position 1000 to 9000 on the positive strand has multiple mRNA exon coding sequences nested within it. I hope I'm not wrong. Totuko, I'm right? Anyways, follow me. I think I'm right.
Okay.
Sorry, did somebody say something?
Yeah, sorry, please can you come again with what you said?
Oh, okay. So I said that looking at this example here, so we are saying that this gene Eden that runs from position 1000 to 9000 has this mRNA exons and coding sequences. So that was what I said. So I was just trying to rethink again that I'm not wrong, right? But anyways, so so basically GFF and GTF are annotation files. You know, just I had already mentioned to you that you want to describe features, you know, you want to describe genes, exons, etc., etc. So for the oh okay, so um so you have, if you look at this same diagram, you can see mRNAs, exons, um CDS. So your mRNA, this different versions of the gene transcript, the exons, part of the gene that become part of the final RNA. The CDS is the coding sequence. I mentioned it when I was talking about GenBank as well. So something you'll be familiar with. So that's part that actually code for protein. So these are positions actually. So 1000 to 9000, 1000 to 1012, and so on and so forth like that. So the GTF is like a stricter sibling of BFF. So the GTF actually means gene transfer format, and it's a file format that is used for representing the annotation of genomic features, primarily focusing on gene structures. Just like I said, it's a sibling of GFF, but it's just more strict. So it uses the same columns like this. I mean, columns like this. Um, but it handles how should I say it? It handles more with rigidity. So so the GTFR consists of one line per feature, with each line containing 9 columns of data. The first line can be a comment starting with hash, or this and that. Well, I don't really use the GTF files, so but I'm more familiar with the GFF files. So yeah, so that's like the summary of the GTFR. So if okay, so we can do a quick, just to reiterate some of the things just briefly. So we will start from GenBank. So GenBank actually, we said, it stores all the information, everything, all the information in this world. So, and you can find it in databases, archives. So faster, you have your sequences, you know, used in for alignment, for your reference data. You have you have your FASTQ, so you have your sequences plus your quality score. Remember, it has 4 lines, so that's like your raw read. And you have the fact we talked about the fast QC, which is your quality summary. So it tells you how good, how bad, your data is before you proceed to your downstream analysis. And then we went further to the soundbar BAM. So it starts read up alignment. So and then from there we went to VCF variants, and then last but not the least, the GFF and the GFF GTF, so which are annotation, so features and annotation. So you use them in gene structure, RNA-seq, etc., etc. So I would say that every format we've spoken about today is like a puzzle. I know I've mentioned puzzle like 3 times today. It's like a puzzle. So when you combine every one of them, from your sequences to your alignment to your variants, your annotations, and you when you combine all of them, you can actually reconstruct um a story of a genome, the story entire story of a genome. Like when you construct every, I mean, when you reconstruct everything together, it's like when you make a puzzle, everything is scattered and you fix it and then it tells you a whole picture. So that's how all these formats are. So if you take, so I would say that when if you understand your file format, you understand your data. So if something goes wrong, you know where to go to, you know what to check, you know how to open and read and interpret. Oh, okay, this must have been where it came wrong, and so on and so forth. So thank you as you manage this lecture that I have given to you. Mm.
Okay, yeah, thank you very much, Kemi. Thank you so much. So please, do you have questions before we go off for the day from anybody? No questions. Um, okay, I guess not. So I think that will be that for today. Kemi is no longer here too. She just left. Please, can you go ahead, Samuel?
Thank you. Um, my question is that the BAM file, um, I like to know, is the BAI file generated along with it? Um, I think your slide says the BAI file contains the table of contents for the BAM file. Hello, can you hear me?
Oh, she just left the call again. Let me see if she can. Samuel, please, can you come again with your question?
All right. I mean, the BAI file, I think the slide says it contains the table of contents for the BAM file. So I'd like to know if the BAI file is also generated alongside the BAM file.
Sorry, someone keeps
Okay.
calling.
So he's asking if the BAI file is also generated along with the BAM file. That's BAI. Hope I'm correct, Samuel. BAI, not BI.
Oh, yes, BAI.
BAI. Yes,
You
along.
actually index it yourself, if I'm not
Uh,
wrong. Yes, you index it yourself.
uh, okay, so if the BAM file is a machine readable file, I mean binary. So why do I need to index it myself? I'm not the one reading
She just left again.
Sorry, to answer your question, Samuel. So, I mean, when we do mapping and alignment, So what we usually do first is to first index your reference. So just imagine your textbook. I mean, at the back of your textbook, you usually have like all this index section, and you notice that if you look, let's probably you are looking for a word, any word, and you go to the index section, so you will see all the pages that you can find all of those words. So instead of probably just so let's probably you don't have index and say probably you are looking for a specific word, it's going to take you a long time to find a particular, the page where that particular word is. But with index, so we just go to that the particular page, and then you can locate the word very fast. So that is the same analogy that indexing bring to the table when we are doing alignment and mapping. So you first index your genome, your reference genome, so that whenever you want to map to any particular location, you can just look at that particular. So instead of probably just searching exhaustively, it's just going to go to that particular location and then map that particular route. So that is actually what is going on. And when you index, so what you get is a uh, .fai, and when you now probably it's also usually very large. When you now compress it, so you get by. So I'm not sure whether you understand, but the idea is just to, you indexing your genome is, just breaking your genome into chunks and then indexing the way this particular K mass or of particular length. So this is where you find it. This particular K mass, so this is where you find it. So things like that. I'm not sure whether I answer your question, but just imagine like a textbook, how you index your textbook. So that's actually what is happening when we index our reference genome before mapping.
Okay, thank you very much. Please, I have another one. The file formats you've examined today, I think they are basically on the genome, the nucleic sequences. Are we getting anything on amino acid sequences for protein, others? Maybe
Yeah.
their sequences or their previous structures.
Yes, for amino acid sequences, majorly the way we capture them is we either capture them, using the dot faster, faster formats. And so that's when she was talking about. There are so many ways that you can represent your faster formats. Dot FNA is for nucleic acid, and dot FAA is for amino acid. So that is one way. So you can organize your faster sequence, your amino acid sequences in a faster format or in a gene bank format. So those are the two popular ways where on how people actually organize their amino acid sequences. So the one I'm very familiar with, the gene bank and also the faster formats. I don't think there is any other formats that you probably can use to represent your amino acid sequences aside from those two. I might be wrong, but those are the two popular ways you can actually use to store your amino acid sequences. So yeah. Does I'm not sure whether that answered your question.
Yes, it does.
Sam.
It does. Thank you very much. Thank you very much.
Yeah, thank you very much. So we have one in the chat box here from Akintayo. So should I read it out or you want to unmute and speak? So you're asking about FASTQ file.
Okay. So I'm not sure that you can read it. My tab
Okay,
is not
so
responding.
Akintayo is asking, so for the FASTQ file that has for a FASTQ file that has a very poor quality score, I'm taking that the file is not good, right? So I think it's trying to say that can we compare search through other base search through other databases to sort out the poor quality score that's for the process file. So like I think from what I understood from your question, like each um maybe output you get from your particular why can't you take your quality score specific to that particular domain that you want. So like it's not as if you can just pull it up and start comparing it to what you have in other databases. It's either you use other tools to probably maybe for example now you can trim your rates and do like a whole lot of other stuff. You see if you can maybe like kind of improve the quality of your rates, or you can maybe like go back to maybe preliminary steps to, to do either to sequence the gain or maybe like any other method which is not coming to mind now. But like you don't really like each output you get is specific to that particular genome, to that particular organism that you frequent, and then was able to get it fastly for. So you're not comparing it to a computer when you're trying to sort it out. Maybe like use other tools to try to enhance the quality of the. So I don't know if you have any other thing to add to that.
Yeah, I think I understand the perspective is coming from. So what you said, it is true. Sometimes some people use that. I won't lie to you. So when you assemble your genome, so probably when you get your FASTQ files and you assemble. So what most people do sometimes, I mean, one way that you can probably assemble is to do, I mean, you know, there are two ways. So we have de novo assembly, we also have reference-based assembly. So when you have like a reference that is well polished and then you sequence, so you can just probably compare what you get. I mean, your sequence that, I mean, that are of poor quality, probably map it against the correct one just to see and correct. So that is one way. But most of the time, we are always interested in. I mean, scientists are always doing de novo assembly. So you want to detect something new. So because sometimes your reference can be biased, so we know we have a lot of variations in organism. So organism like shows a lot of variation. So when probably the reference that you now have is just like a specific version of a variation, and now when you are now trying to like probably correct, I mean, the new sequence that you have that might be of another probably fashion to the reference. So sometimes you might not actually probably get the true picture of what's actually going on. So sometimes we don't. I mean, these days most people don't actually do reference-based assembly. So they do the novo assembly. So if you are doing the novo assembly, so that's when we start doing all this quality check. And once you are doing that one, so I mean, you don't want to be comparing to a reference because you already know that your reference is going to bias the results that you are having. So just imagine probably, so let's probably you have like three variations in the population, and the reference that you have is now of like a, let's say probably we have like it belongs to like a fashion C, and now you are sequencing fashion A, and now you're trying to like compare, I mean, correct the inefficiency in fashion A using fashion C. What we still get is what is fashion C, whereas what you are interested in fashion A. I'm not sure whether you get the analogy. So that's why we're always interested in the novo assembly. And when we are doing the novo assembly, we don't want to be, I mean, using other databases to correct for mistakes. I'm not sure whether that answered your question, whether you understand the logic of what I'm trying to like talk about. Akintayo. Sorry, I can't hear you.
Can you hear me?
Yeah. Can you speak louder?
Uh,
Hello?